right, guys, we're going to talk about Chapter 3. This is Metabolism, and so let's go ahead and get started. We are looking at a picture where we have a cell, a cartoon with all different kinds of activity going on, processing nutrients, creating energy, and that's basically metabolism. So metabolism is the sum of all chemical reactions that occur inside your cells. They include catabolic reactions, which is the breakdown of larger molecules to smaller ones, and anabolic reactions, which is opposite. It's creating large molecules from smaller ones. And we'll also talk about energy metabolism, how energy is stored and used inside your body or inside your cells. So one thing to remember or to understand is chemical reactions. So we have our reactants before the arrow. The reactants can also be called substrates when you're talking about enzymatically controlled reactions, which basically everything is inside of our cells. The arrow denotes the process of the reaction, and then our products are on the other side of the arrow. Now, it's important to understand that arrows can be bidirectional. So if you look at an arrow like this, right, we have an arrow going forward and backward, which means that the reaction can proceed in both directions. The reaction can proceed in a forward manner, where the reactants become the products this way. Here are the products. Or it can be reversed, where the products are the products are transformed into the reactants. So that's a bidirectional. So for a bidirectional reaction to occur, the enzyme that catalyzes that reaction is able to bind to both the reactants and also to the products. All right, we also want to understand a metabolic pathway. So a metabolic pathway is where you have a series of steps to get to your end product. Almost all chemical reactions in your body have metabolic pathways involved. The first molecule in a pathway is called the initial substrate. The last molecule is called the end product, and everything in the middle are called intermediates. And where are enzymes written? Enzymes are written on top of the arrows. So you'll find the enzyme's name written out on the arrows, or sometimes you'll just have a number, right, telling you that there are different enzymes involved in the process. All right, so let's take a look at our metabolic reactions. Our metabolic reactions, this is a little bit of a review for some of you, so we'll go through each one pretty quickly. So the first one is condensation hydrolysis reactions. So these are opposite reactions. A condensation reaction is going to create larger molecules. So the reaction that takes place between our two monomers here, and these are monosaccharides, right, is going to link up those two monosaccharides, and we're losing a molecule of water in the process. So condensation is also known as dehydration reactions because we're losing water, right? In the opposite reaction, we have hydrolysis. So we're using water, a molecule of water, to break up that bond between our sugar molecules there, and we're breaking the bond with water. So hydro refers to water, lysis refers to breaking, so we're splitting up molecules into the smaller units. The second D is phosphorylation. I'm referring to your outline, by the way. D is phosphorylation and dephosphorylation. So this is where you add a phosphate group. So remember, a phosphate group is PO4. It's not just a phosphate. So you see we have a substrate. We are going to add a phosphate group to that substrate. The enzyme that's used in this are called kinases. Okay. Now we can also take off the phosphate group, and another enzyme does this, and they are called phosphatases. And so these are just a group of enzymes. There's lots of different kinases, there are lots of different phosphatases, but that's what we call. So kinases add the phosphate group and phosphorylate something. So here we would say that substrate has been phosphorylated. We take the phosphate off, and now we have dephosphorylated that substrate. In oxidation reactions, so I like to always think about this mnemonic, Leo the lion goes grr, where a compound, if it loses the electrons in the chemical reactions, it's said to be oxidized. If it gains electrons in the chemical reaction, it's said to be reduced. So let's look at this picture here, where A and B are having a chemical reaction. A is transferring its electrons to B. So A has lost the electrons. A is oxidized, right? A lost the electrons, it is oxidized. Whereas B is the one that gained the electrons, so B gained electrons, it is reduced, okay? So whenever you see chemical reactions occur in the cell, these electrons are oftentimes associated with the hydrogen ion. So when you see hydrogens being passed back and forth between two molecules, these are redox reactions or oxidation reduction reactions, right? Redox is the shorthand, reduction, oxidation. An example of a very long pathway, metabolic pathway, is cellular respiration, right? Cell respiration, where you have glucose, and glucose is slowly oxidized to carbon dioxide. So glucose is losing their electrons, and then CO2 is gaining the electrons. I'm sorry, not CO2, gaining the electrons. Your electron carriers are gaining the electrons. And then ultimately oxygen is going to get those electrons. Sorry. All right, let's move on to answer this question. Metabolism is? So you can pause. Every time you see a question, you can pause it to think about it. If you want to think about it, try your hand at guessing the answer. But I'll always tell you the answer, all the reactions in the cells. What is the opposite of hydrolysis? And it's condensation. All right, so energy requiring reactions are called endergonic. Endergonic, the term is for energy requiring. An example of energy requiring reactions would be anabolic reactions, where you build up larger molecules from smaller precursors, such as building proteins. So amino acids are put together to make a protein. This is an anabolic reaction that requires energy. There is a change of potential energy from the reactants to the products. When the reactants have less energy than the products, this is called a non-spontaneous reaction. So if you think about a slide, if your reactants have a lot of energy, then the products, after the chemical reaction happens, has lower energy. This reaction is favorable to happen because molecules like to be in a low energy state. Whatever the lowest amount of energy is, that's the preferred state for something to be in. So this is what we call a spontaneous reaction. But if it requires energy and the reactants have less energy than the products have more energy, this is called non-spontaneous, because this is not going to happen in the physical world spontaneously, right? This is going to require energy input. It's going to require a lot of work to do this. So that's what we mean about not spontaneous. And the products have more energy than the reactants. And so energy is put in, and that energy that's absorbed is going to be found stored in the products. All right, so let's talk about activation energy. So the activation energy of a chemical reaction is the energy hump that a chemical reaction has to proceed. In this example, we actually have a spontaneous reaction. We have the reactants have higher energy than the products. And if we didn't have that activation energy barrier, the reactants would just become products. There's nothing holding back the decomposition of the reactants to make the products. However, we always have activation energy barriers. So think about if you have like a large molecule with a lot of energy. Let's say we have chocolate, a nice chocolate bar, right? Lots of fat, lots of sugar. if you have a chocolate bar in your cupboard, it doesn't spontaneously break down into glycerol and fatty acids, and the sugars in there don't break down to their monosaccharides. The chocolate is really stable. So all those organic molecules are stable because in order to break them down chemically, each one of them has an activation energy barrier that holds back that spontaneous breakdown. So this activation energy barrier is really important so that large energetic molecules can exist in the world without spontaneously breaking down. However, if we want to break things down, we need these activation energy barriers to be obliterated. We need to knock it down. So enzymes are the tool that biological systems use, right? But living things use to reduce the amount of activation energy required for that reaction to occur. So with an enzyme, okay, you still have the same energy in the reactants and the same energy in the products, but with an enzyme, you're going to have a much smaller activation energy barrier to get over for the products, for the chemical reaction to happen to create products. So what's happening is we're reaching a transition state. So at the very top of our activation energy hump, what is achieved is this transition state. The transition state is a state where the bonds between our reactants is unstabilized or destabilized, or that they've absorbed so much energy that they're very reactive. And so this is the pinnacle, this is that halfway state, that transition state right before we become products. So we're either destabilizing bonds that exist, or we're giving individual molecules enough energy to make the transition to products. So enzymes can reach that transition state with less energy involved. So what they do is they reduce the amount of activation energy and can reach that same transition state with much less energy. So let's look at, right, so just to look at some terms here, the transition state, the reactant and product transformations are not sudden. They have to reach this transition state by absorbing a lot of energy. This energy is the activation energy barrier, right? So that energy barrier is called activation energy. And this is going to limit how fast a reaction goes. So if you have a very high activation energy, you're not going to have a very fast reaction. And so in order for reactions to proceed at the speed that living things need things to happen right away. So enzymes are used to reduce the activation energy so that reactions can happen very quickly. All right, so let's see if we can answer this question. The answer is low, right? If activation energy is high, then the rate of reaction is low. All right, so what do enzymes do in the process of chemical reactions? Enzymes are proteins, all right? And they are catalysts for reactions in biological systems. Catalysts, by definition, increase the rates of chemical reactions, right? That's what, in chemistry, if you do chemistry, that's what catalysts do. They make reactions go faster, right? Increase the rate, make it go faster. In your cells, the only way you can get reactions to go faster is to decrease the activation energy for those reactions. In chemistry or outside a living system, you can warm things up. You can add heat, put fire to it, and then you can actually get these chemical reactions to happen much faster. But you cannot change the temperature in a living thing. So living things require enzymes to lower the activation energy. So enzymes will bind to a reactant. The reactant is called the substrate, and then form a complex. This is the enzyme substrate complex. And after that complex has formed, it will spit out the product and enzyme. So let's take a look at this this process right here, right? So this is a quick review of enzyme action, where enzymes have an active site. The active site is where the substrate binds. Here's our substrate. The substrates come in, and this is our enzyme substrate complex. This is what reduces the activation energy, because when the substrate's bound to the enzyme's active site, the active site tweaks the substrate in a way either by holding two reactants together in the proper orientation so that the chemical reaction can happen faster, or it destabilizes, it pushes and pulls on the substrate and destabilizes certain bonds, and so the chemical react bonds break or bonds get put together. But this complex is not permanent, right? This enzyme substrate complex is done by weak bonds because we don't want a permanent substrate in the active site. The substrate then either turns into the product, or sometimes the substrate just bounces back out, right? And so then it's free to come back into the substrate. The product, right, if the substrate has turned into the product has less affinity for the enzyme, and the product will leave the active site of the enzyme, and then the enzyme is ready to be used again. So this is a good place to talk about how enzymes are reusable in chemical reactions. They are not destroyed. They are not altered in any way. They're recycled over, and they can be reused over and over again, thousands of times in a second sometimes. So before we move on, let's talk about ligands and affinity. So a ligand is any molecule that binds to proteins. So substrates are ligands, and ligand binding is always by weak interaction, which means hydrogen bonds, ionic bonds, anything that's not covalent, because covalent bonds are basically permanent and they're very strong bonds, right? So the affinity is the attraction of these ligands to the protein. So when we talk about enzymes, it's what's the attraction of a substrate to the enzyme's active site. So in this picture, we have two ligands. We have ligand A and B, and we have a protein here with a binding site. Both of these ligands will fit into the enzyme site there, but one fits with greater affinity, right? So if we take a look at the graph, if we look at at any concentration here, right, we have the binding of, so this is the number of bound ligands. The binding of ligand B, it binds, but at a lower rate or lower number than ligand A. So ligand A has a higher affinity for that protein. It's going to just end up binding there more often than ligand B. And if you look at what's happening here, right, ligand A has 3 positive charges close together. Your protein has 3 negative charges close together. So that's going to be this ionic attraction that ligand B just doesn't have a strong one, because ligand B has a negative charge here, which is actually going to repel from this negative charge here. And then it has these positive charges, which would repel from positive charges here. So even though perhaps this positive charge can be attracted to the negative charge, there is some weak attraction, but just not very strong. Ligand A has the higher affinity and therefore will bind to the protein more often. And this is a key concept to know about just how things bind to enzymes, like what would cause enzymes, the substrate to bind to an enzyme or anything to bind to a protein. It's the affinity. All right, so let's look at some other ideas here. So let's look at the lock and key and induced fit. So substrate specificity. We know that enzymes are specific. Let me talk about this one, right? Enzymes are specific for one set of substrates, or it could be a similar group of substrates. So molecules that look similar to each other. Enzymes are not changed in chemical reactions. Enzymes are not consumed in a chemical reaction. Like I said, most recently, they can be reused. And enzymes are identified by their suffix ase. And they're oftentimes named after their substrate. So, for example, this sugar lactose is broken down by the enzyme lactase. So look how similar those words are, right? So the enzyme, you know it's an enzyme because it ends in ASE, but it's named after the substrate that it works on. Another good example is peroxidase, which catalyzes hydrogen peroxide. I was trying to think of another one. I can't right now, but anyways, you get the idea. All right, so let's take a look at the two models. So how does the substrate fit into the enzyme's active site? There is a lock and key model, which is basically where you have a active site that is sort of fixed in place. You have substrates that the shape doesn't change either, and then whichever substrate it fits into the active site, like a lock and a key, that's the substrate for this enzyme. The problem with lock and key model is that enzymes, most enzymes are reversible. Let me just add our reversible. So if there's one enzyme that catalyzes A plus B to the product C plus D, for the lock and key, right, the active site should only fit the reactants A plus B and should not fit the products C plus D. We spit the products out. But in fact, most enzymes are reversible, which means that C plus D, the products, should also be able to bind to that active site. So if you have a reversible reaction, the enzyme should be able to fit A plus B as well as C plus D. So that brings us to the induced fit model. The induced fit model is basically defined by the active site approximately fits the substrates, but after the substrate binds, there is a conformational change. What the word conformational means is shape. Okay, so there's a shape change in the active site that allows it to fit better. So it kind of snuggles up with the substrate, and then the reaction takes place. So it looks kind of like this, right? Here's our substrate. This is our active site. Doesn't look like it's going to fit, but after the binding happens, the enzyme changes its conformation to be more well-fitting to that substrate. We have the reaction occur, and then the product has less affinity, and so it'll spit the product out. But this can be reversible. This can come back in. You see these double arrows. That means it's reversible. And then the reverse reaction can take place. All right, so that's that. Let's talk about cofactors and coenzymes. So cofactors and coenzymes are molecules or ions that are binding to the enzyme to help it work more effectively. Metals are your cofactors, okay? Metals will bind to the enzyme and usually allow the substrate to bind to the active site with higher affinity, therefore allowing the chemical reaction to happen. Coenzymes are vitamins. They are organic molecules. Okay, so that's our big difference. Cofactors are minerals. They are inorganic. Coenzymes are organic molecules. They are derived from vitamins. Here are some coenzymes: NADH, FAD, Coenzyme A. These should look somewhat familiar to you because these are all used in cell respiration. Okay, so we need to take our vitamins and cofactors. I would say vitamins and minerals, right? So your enzymes can work efficiently. All right, so enzyme catalyzed reactions. Factors that affect the rates or how fast enzymes can catalyze reactions. How many molecules? So when you look at the catalytic rate, when you look at the enzymes, how fast it works, you're basically looking at how many molecules are generated per minute or per unit time. This can be per second. So you're looking at the amount of product that's being put out. Some chemical reactions, some enzymes can generate thousands of molecules per second. Others may take a minute or two, more than a minute, to catalyze one molecule. So enzymes work at different rates. What can affect the rate of an enzyme? So there's a substrate concentration. If you're just looking at how much product is being made, then the more substrate you have, the more of it will fit into the active site of the enzyme, the more product will be made. So this is basically that. And then at some point you reach saturation. So when you have a graph, so this is the amount of substrate that you're adding, right? And this is so a lot of times the graph will look like this. So when you add more substrate, the reaction rate goes up. And then at some point, the reaction rate, this is the rate, it flattens out, it flatlines. at this point, you've saturated the enzyme. So this is the most substrate, or it's the point at which all enzymes are working on a substrate. You can't spit out any more product any faster. So this is called saturation point. Anyway, so the substrate, when you increase the substrate concentration, yes, you can get more product up to a point, up to that saturation point. Another pretty obvious one is how much enzyme do you have? If you want a lot of product, then you can actually control, or the cells can upregulate how much enzyme they're making. So you can increase your enzyme concentration to increase the product. The affinity, right? How well does that substrate fit into the active site of the enzyme? And this is where cofactors and coenzymes can come in to play and affect the enzyme. And then temperature and pH. So we know that enzymes are proteins. And so temperature and pH will affect all proteins, not just enzymes. Higher temperatures will start to denature enzymes, and alteration of pH will also start to denature enzymes. Denature means that the enzyme shape or conformation has been broken down. It's not functioning in the way it's supposed to be functioning. And this is why high temperatures, high fevers are dangerous for us. And then also, if our blood becomes too acidic or too alkaline, it can cause coma and death because our chemical reactions just stop happening. How can we regulate enzymes? So we have 4 different topics to talk about. So allosteric regulation is the fact that you can regulate an enzyme by binding something to a regulatory site. So this regulatory site is sometimes called an allosteric site. Okay? And a modulator, also sometimes called a regulator, is a molecule that binds to this regulatory site. So let's look at that, right? So we have an enzyme, here's the active site, here's a regulatory site, here's our modulator. Our modulator binds to the enzyme, but look what happened to the active site. The active site changed. And this changing, this altered activity can either go up, it can cause the enzyme to work faster and have higher affinity for the substrate, or you can downregulate the enzyme, you can make it less fast, or the reaction rate can be less fast, or the substrate has less affinity. Okay, so you can increase or decrease the activity. It changes the shape and activity of the enzyme. Okay. So here's a nice graph showing you. So if we pick any substrate concentration, notice at this point we're reaching saturation. But so let's just pick a concentration, right? So at this concentration, if we have an inhibitor on our enzyme, our reaction rate is pretty low. The normal enzyme has a higher reaction rate, but if you add an activator, something that activates the enzyme, makes the substrate bind better, or increases the catalytic rate, we have much more a faster rate, right? So activators are for increasing enzyme action, inhibitors are for decreasing it, and your cells will create the activators or inhibitors. All right, let's talk about covalent regulation. This should look really familiar. We are going to add a phosphate group. Okay, so we can have an enzyme in an active or inactive state. When you add a phosphate group, you create an active state usually. So here's an enzyme, here's our chemical group. Most of the time this is a phosphate group. So Enzyme A, you have an enzyme that adds on this chemical group, creates a covalent bond. It's a very tight bond. So in order to remove that bond, you have a second enzyme that takes that off. If this is a phosphate group, we already know the names of the enzymes, right? So kinases are going to put on that phosphate group. Phosphatases are going to remove the phosphate group. Okay, so here it is. Phosphorylation is a very common way that you can control enzymes. Some enzymes are not active until you have that phosphate group attached. attaching by kinases, removing by phosphatases. If you remove the phosphate group, you shut down the enzyme from working. Feedback inhibition is very common in metabolic pathways, and this is where you have some upstream, intermediate, or even sometimes a product feeding back to an enzyme in the middle of the pathway. So in this example, E1 and E3 are constant. They work at a constant level, but E2 is the rate, the concentration of C can influence the rate of enzyme 2. Enzyme 2 is known as the rate-limiting enzyme. So the step from B to C is the rate-limiting step, meaning that this is the slowest, or this is the one that's regulated and controlled. So feedback inhibition just means that C becomes an inhibitor, because we're using inhibition, right? C is an inhibitor of enzyme 2. So as the amount of C goes up, we can start to slow down the process of making C by feeding back and slowing down the enzyme. Here's another example with the end product, right? So there's the metabolic pathway. In this case, the first enzyme is our rate-limiting enzyme, and our product, our end product, feeds back and negatively feeds back to enzyme 1 and inhibits enzyme 1. So this is a good way to regulate a pathway. So if you are always running the pathway, you're going to get a lot of your end product, right? You're going to get a lot of D. So as the levels of D increase, the concentration of your product increase, you might want to shut off that metabolic pathway. You might have enough D for now, right? You don't want any more. So then the end product is going to shut down the whole process. So it's a way of regulating a metabolic pathway. It's a way of not overproducing the product, right? You don't want too much end product. And this is just something for you to think about, though. Some pathways, we talked about these metabolic pathways as being linear, right? But it doesn't have to be. It can be branched. So we can actually have two enzymes work on the same substrate, giving you different products. So this is a branched pathway. Feed forward activation, right? You have a plus sign for activation, is where you have an intermediate that acts as an activator for an enzyme in the pathway. So this means that this might be a pathway that is something the cell wants a lot of the product quickly. And so we're going to have the intermediate. Once an intermediate is created, it helps to speed up the chemical reaction for that last product to be made. Okay. All right, so the answer here should be easy. Letter B, lowers the activation energy. All right, so we're going to pause. That's the end of part 1.